All right, everyone. It is 12 noon by my clock. And Ryan has some fabulous information to share. I want to get to him and give him as much time as I can, so we'll go ahead and get things rolling. I'll do a quick overview of Trees Forever, who I am, what we do. Today, I'm really excited to have Ryan here with us. Thanks, Ryan. This is going to be awesome. Our webinar today is Wood is Good, an overview of tree growth and development. And this was really intriguing for me. It's a topic that I was very interested in, and so we're very happy to have him here. Really looking forward to it. My name is Nick McGrath. I am the East Central Iowa Field Coordinator and a certified arborist here with Trees Forever. The big thing in my life is assisting with tree plantings and conservation work, trying to be eco-friendly wherever we can, wherever I can. I'm a huge advocate for native species, and I really enjoy planting wherever I can, a diverse selection of those trees. Here at my own home, I'm excited to be growing pawpaw and tried my best at hand pollinating this year. We'll see if there's any success. Come back in a couple of months to find out. For my work, I travel through those yellow counties there, working with communities of all sizes toward that ultimate growth of tree canopy using primarily native species. And then Trees Forever is a very well-established organization. Our mission is to plant and care for trees and the environment by empowering people, building community, and promoting stewardship. And we do that through a wide variety of programs and offerings and different events each and every year. We've planted since 1989 over 3.4 million trees. We are a charitable 501c3 organization. We rely on people like you to support our growth and development and our work statewide. We are also in Illinois and pushing up into Wisconsin. We assist over 200 communities and landowners annually, engage an average of 7,000 volunteers, and award over $650,000 in grants with technical assistance. We love going out into communities and helping with any and all of your tree needs. Please reach out to us. And we have a bunch of upcoming events, just a couple to highlight here. Currently, we're rolling out our Stewards of the Beautiful Land program, and that has two different segments. We have bi-weekly webinars on quite a range of topics, and then we have several in-person field days, 4 each in Blackhawk, Des Moines, Harrison, and Story counties. We also have tree keeper workshops going on right now in Iowa City. Links to both of those are there. Our next tree keeper session is Saturday, June 1st. We'll be discussing tree care and maintenance with Stephen Carlson, and then we'll be rolling out those same programs in Des Moines and Cedar Rapids this fall. Please look out for those dates. Check back with treesforever.org for all of our events, all of our happenings and opportunities, and contact information for myself and a field coordinator that could serve your community. Without any further ado, I want to turn this over to Ryan. Give me just a moment. I'm going to let you go ahead and take your own introduction here.
Great. Well, hey, thanks, Nick, and for the invitation, and thanks everyone for being here. My name is Ryan Murphy. I am a researcher and educator at the Department of Forest Resources here at the University of Minnesota. More specifically, I help manage the Urban Forestry Outreach and Research Lab. So we do a lot of teaching, outreach around the state, talking about trees, tree ID, plants, pests. We also organize the Minnesota Shade Tree Short Course, which is sort of a cousin conference to your Iowa Shade Tree Short Course. And I also have a personal research interest in finding survivor elm trees that are resistant to Dutch elm disease. So we have a elm selection program here, and so we get to go around the state looking for big old elms and grow them up and inoculate them with the fungus to see if they can survive. But that's just a little bit about me. Let's jump into this one here. Oh, I should also say, when I sent this title, Wood is Good, over to Nick, it just all of a sudden, Wood is Good started popping up all around me, like people with bumper stickers, YouTube videos with the same word. And so if you're here and Wood is Good is your trademark, my apologies, but it's a good phrase. So why don't we get into this with this question here. Why does a leaf grow the way it does? Or said another way, why does a leaf look the way it does compared to another leaf? This was a question that a friend and I had during plant propagation, the first plant class I took here while I was an undergrad with Jeff Gilman. And I can't quite remember what he said, but he had some good responses, like that's probably something about the auxin. But he also left it open in that we don't necessarily really know. One hypothesis might be that it's all just genetics. So plants, like all other living organisms, have a genetic code that codes for different proteins that can make molecules and all sorts of things. Here's just a little snippet of the genetic code, which codes for the chlorophyll molecule, which we'll come back to. A little bit more about DNA and genetics, because I'm a bit of a plant nerd, so I think it's interesting. So DNA is this chain of nucleic acids, and then that DNA gets bundled up into what we call chromosomes. We've got them. We've got chromosomes, and then we've got a copy of our chromosomes. So we're what you would call a diploid. So we have two sets of our chromosomes. Well, plants have all these different ploidy levels, call them polyploids or just ploidy. So here's some different selections of ginkgo at different ploidy levels. So you can have these kind of weird plants that are haploids. They just have one of their chromosomes, triploids, all the way up to tetraploids. And this will keep going for plants. For example, I think wheat is like a hexaploid, 6 copies. You can have octaploids. I'm not really sure what the limit is, but just one last thing to point out here. You can see on this one, this little haploid number 1, and if you compare that down to number 32 here, the tetraploid, it should be pretty clear that there's a fairly large size difference. And that's one of the features of having these larger polyploids is you get bigger plants, bigger fruits, more production of metabolites. Okay, so we're working from this, we're building a tree. And so we're starting with some DNA, right? And now, so we've got a plan. Now we need some building blocks, right? We need some energy and building blocks. Just like if we're going to build a house, we've got a blueprint. Now we got to get some materials. We got to get people to help us build. We got to have money to pay the people to build the house. So trees are autotrophic, which means they make their own energy. And compare that to ourselves, we're heterotrophs. So we need to get energy from other things. Same way with fungi, right? They need to break down elements and then absorb it into their body. Well, trees are autotrophic in that they can just use the sun's energy to create the food that it needs to grow. So here's photosynthesis, and we could do a whole talk, probably, I don't know if I could do the talk, but if we really wanted to get into the biochemistry of photosynthesis, we could. But what I wanted to do is try and distill down what I see as the most simplest structure. So you have a carbon dioxide, you add a water molecule to that, throw in the energy from the sun, and then you have the output from that reaction. You get a sugar and an oxygen, right? And so this oxygen component comes up quite a bit when you ask people, why do you like a tree? And they say, oh, it gives us air to breathe. And they're the lungs of the earth, right? So, but what are we going to do with all this sugar? Well, you could take a simple sugar molecule and turn it into a polysaccharide. Polysaccharide is just a way to say a chain of simple sugars combined together in different formulations. Here on the left, you're looking at starch. So trees, they're going to use starch as an energy storage molecule and then be able to utilize that later. Glycogen, that's what we can use in our bodies. So our livers and muscles can store glycogen. And then another really important one for plants on the right is that cellulose molecule, polysaccharide, that is used in constructing cell walls. So we hear a lot about trees give us oxygen and they photosynthesize, but the other half of the story, and very important part of the story, is that they're also doing respiration. So they're basically, you can take that photosynthesis equation and just flip it, right? So now you take a sugar and add an oxygen to it, and then through the magic of the mitochondria, you then get carbon dioxide, water molecule back, plus you get the energy here in the form of ATP. And so, you know, trees need oxygen too, and respiration is critical for all living things. If respiration stops, you know, the organism dies. So very critical. A little bit more dealing with this structural component, the cellulose. So this is what plant cell walls are made out of. So it's this combination of cellulose, hemicellulose, and then it gets bound together with something called lignin. And these sugars, the cellulose and the hemicellulose, that's really good stuff. We want that. We want to make paper with it. We want to extract it and make biofuel with it. Insects want to eat it, but it's actually kind of tough to get at because the lignin is in there, and the lignin is a waterproofing molecule and also provides really good reinforcement. I would just say one other thing here is that I thought it was interesting in preparing for this talk to find out that plants don't actually have enzymes to break down cellulose. So once it makes it, it's kind of there. But then you'd have like a decay fungi, which has the mechanism to go in and utilize that for energy. In trees, we can think of growth as having a source and then a sink. So for example, leaves are photosynthesizing. They're creating that energy, creating those sugars, which then are transported to different sinks or parts of the plant. For example, areas that are growing like the roots. So growth arises out of a couple different meristems. Here's an image of an apical meristem. So meristems are groups of cells. They're just undifferentiated cells. You might call them pluripotent. They can turn into other things. You know, we have our stem cells. That's what these meristems are. So really critical for the plant in terms of where that growth is coming from. So this is where, for example, roots can come from apical meristems, and the shoots come from apical meristems. And then we also have in trees a lateral meristem, or what we call the cambium, but we'll come back to that one in another few slides. So the tree, as you can imagine, if you think of, you could imagine the meristems of being kind of like buds, right? So there's a apical meristem there and a bud, and trees have all these meristems that are growing. And I think this is kind of an interesting thing that can happen with trees is you can get mutations, right? You can, you know, our cells can mutate, and that can lead to poor consequences. But in this case, this somatic mutations can actually be beneficial to the tree. And so you have a branch that's mutated, and now you have a new genotype. We'll call it genotype, but it's a genetic chimera within the same tree. So on the right, it's a tree that was found. It's a eucalyptus tree, and the photo's grainy and tough to see, but this area here in the top left part of the canopy, that's all healthy leaves. The rest of this is all being defoliated by an insect, and the researchers were able to go in and show that that part of the canopy had mutated and so now it's got a different set of gene expressions, and then also that then coded for different compounds that provided, in this case, a benefit against insect herbivory. So sticking with growth here, so new shoots and leaves are coming from buds, right? So the magical world of buds, we won't go too deep into it. That's probably more for a tree ID type of course. So just pointing out here with this photo, this point where the apical bud broke and now is growing this new shoot. And right here in this point, there's what we call the bud scale scar. And that scar will remain on that twig, and you can see successive years of bud scale scars. And so it's a nice way to get a sense of how a certain tree is doing in terms of its vigor, or if it's been experiencing some stress, because you're either going to have more growth or less growth in a given year. Another thing to keep in mind is as those leaves are emerging, right? So we've got a dormant bud, then they push out into a leaf, but that leaf needs energy to actually produce its structure. It's not photosynthesizing yet, it has to actually grow. And so there needs to be that stored energy in the twig or in that bud that then can produce the leaf. So in that case, the starch within the twig was the source, and then the leaf became the energy sink for that. Thinking about light absorption and a little bit about leaf biology. This is just a cross section of a leaf. You can see all the chloroplasts in there. That's where all the photosynthesis is happening. Another important part of leaf biology is the stomata. The stomata is on the bottom side of the leaf, and these are little pores that can be opened and closed. And this is where that gas exchange can occur, either bringing in CO2 or oxygen. And then this is also critical because this is where water can transpire through. So plants have chlorophyll, and chlorophyll absorbs light energy at 2 pretty specific frequencies. That's why if you ever see like a grow light or like an LED greenhouse light and it's purple, that's because it's really just tuning in on the frequencies that the chlorophyll is going to utilize and not waste other energy. So this is the absorption curve of the chlorophyll molecules, and you can see there's not much absorption happening here. A little bit, but for the most part, it's not using like these green, yellow parts. One other interesting thing I think about this light sensing and absorption. So light's on the electromagnetic spectrum, and just beyond this, so this is our visible light, and then just beyond what we can see, the near infrared. Plants don't necessarily utilize those frequencies for energy, but they have the ability to sense it. And so they do this function where they can tell the ratio of red light to near infrared. And so they absorb the red light. So if a plant was growing in the shade, there's not going to be a lot of red light coming because it's being absorbed by everything above it. But plants don't absorb the near infrared. And so that balance, that ratio, so there's going to be much more near infrared compared to how much red light, and then that can trigger signal cascades within the plant that would lead to elongation. So looking now at the stems, or specifically more the tree trunk, we could start here. Now we're talking about that lateral meristem, the vascular cambium, again, undifferentiated pluripotent cells that can then turn in to the other plant parts. So as those cells from the cambium start to divide to the inside, they produce the sapwood, or known as xylem, some xylem cells. This is the water conducting tissue. Then on the outside, the cambium divides and turns into the phloem. So phloem is living. Xylem and sapwood is, once those cells are created, it's not living. But phloem is living, and this is what's moving the sugars from the leaves to the other parts of the tree. And then the cork, or the bark, also has its own meristem, the cork cambium. And then on the inside, what we refer to as the heartwood. So as the wood stops being conducting, then it just kind of becomes support. And then also the tree will put molecules or antimicrobial compounds into that wood to help reduce decay. Looking at cross sections, this is really nice work by Ziminska, and a nice article, Wood Under the Microscope, published in Arnoldia. Some really nice microscopy work. And so when you have these plant cross sections, you actually need to stain the tissue so that you can see these different plant parts. So here on the left is the unstained, and then by putting stain, you can see the different cell types. So those xylem water conducting vessels are here in blue. In pink, the parenchyma, or might refer to it as the parenchymal rays. So this is part of this is living tissue again, and this is a way that trees can move and transport nutrients in and out of the stem laterally. And then finally here in green are these fibers. So again, these are made up of that cellulose, hemicellulose lignin, but they don't necessarily conduct water, but they're forming rigidity and structure to the tree and kind of making wood what it is. I do want to mention this difference between tracheids and vessels. So vessels, we were just looking at, I believe that was American beech in that last slide. And so that's a deciduous tree and an angiosperm. And so these vessels are only found in angiosperms, and conifers, for example, is a gymnosperm, and they're made up of these tracheids. And, you know, they're performing similar functions. Tracheids are a little bit thinner. I'm not going to go too much into it. I just think it's worthwhile pointing out that those two things exist. So I mentioned gymnosperms and angiosperms. So gymnosperms are evolved earlier, so they're older. They evolved somewhere in the range of 300 million years ago. And the big difference here is that they produce these naked seeds. This is things like conifers. Ginkgo is actually a gymnosperm. Here's tamarack. It's actually a deciduous conifer. And then angiosperms evolved after gymnosperms. And these are a lot of our plants. Here's a cacao. So these are plants that produce seeds in fruits. And it's a very diverse group of land plants. Here you can see 416 families. So within angiosperms, we can continue to look at the vessel arrangement of these species, and there's a lot of diversity in the way that vessels get arranged within a growth year. So on the left, we're looking at what we would call ring porous, and this is a honey locust. And ring porous species will put on these very large vessels early in the growing season. So like right now, they're producing these large pores, vessels. And then as the growing season goes on, they transition and start making smaller vessels. And this is something that we point out because some of the major tree pests that we're concerned about affect ring porous species. So oak with oak wilt is a ring porous species. Elm, because of Dutch elm disease, elm is a ring porous species. And then ash, the emerald ash borer, is also ring porous. And then there's diffuse porous. So here we have a tulip poplar. You can see much more consistent size throughout the entire growing season of those vessels. So in this case, we've got the large early wood and then the smaller pored late wood. So this whole thing here is 1 year's growth. And here in these diffuse porous species, a little bit more difficult to see, but you can still discern the annual growth ring. So we've been talking about the xylem vessels, and these are critical for moving water within the tree. And it's sort of an interesting thought experiment to think about water moving through a tree, right? There's no gas-powered pump down there that's putting pressure into the system and pushing water up the tree. So you have to get water moving in a column 100 feet into the air, for example. And there's a couple of beneficial features of water that help this become possible. One is this idea of cohesion, and the other adhesion. So cohesion is the tendency for the water molecules to want to kind of stick together, right? So you get droplets. Adhesion is when the water kind of will grab on to another substance and kind of pull up a little bit, like a graduated glass cylinder with, you know, that meniscus. So the water kind of gets pulled up the sides. So within living organisms, we have different gradients. We can have, you know, pressure gradients, chemical gradients, and so we're using this, or the trees are using this water potential gradient to move water passively from the ground up through the tree. So we have in the roots, you have higher mineral concentration within those cells, which then creates osmotic pressure. And so you get, or creates a negative osmotic pressure compared to the water in the soil. And therefore water is going to move across that gradient because water wants to move from areas of high pressure or high concentration to areas of low concentration. If we think again about those stomata, those openings in the leaves, so that the air pressure, the vapor pressure just outside of the leaf is a little bit lower than inside the leaf, which means water is going to want to move from inside the leaf to the outside of the leaf. And then as you work your way down back into the canopy, you actually have an increasing water potential gradient. So the water potential down in the roots is higher than in the stem, which is higher than the branches, and then so on and so forth. And so water gets pulled up that column. And one way this can go wrong is if you break that column. So if you had a, call it cavitation, or say you have like a water embolism in that vessel and you lose that column of water, it's very difficult, or I don't know if I want to say it's impossible, but you can imagine how difficult that would be to reconnect that column, and so you would potentially lose that. So speaking of roots, where are the roots in trees? So this is an activity I like to do if I'm ever teaching a class to youth going into a school, as I would start by saying, hey, I want you to draw me a tree, a big tree, and I want you to also draw the roots, and just to see how they're conceptualizing where the roots are in the soil. And we've seen images where you have a tree, you know, a graphic where you have a tree, and then it's basically a mirror image of that tree down below. In terms of mass, that's not necessarily incorrect. You can have up to 50% of the biomass of a given tree in its roots. But where they're found in the soil is going to be closer to the surface. Most of the roots are going to be in that top foot to 16 inches, right? Because there you have good oxygen diffusion. Roots need oxygen. There's going to be good moisture. And you will have some roots that will sink down, but they'll primarily be higher in the soil, and then they can also extend quite wide, up to twice the width of the mature canopy. So within the roots, it's these fine roots and root hairs that are doing the heavy lifting in terms of actually absorbing minerals and nutrients and water from the soil, and then moving them up through the structural roots into the vasculature.
I got a question for you, Ryan.
Sure.
As we're talking about water movement and roots, we have a question from Greg. Do trees gather water through leaf, branch, or trunk absorption? Or is there a way for another structure of the tree other than roots to manage some of that task of water absorption?
That's interesting. I think you potentially could get a little bit of water being absorbed through stomata. Like say you had a very foggy day, but I think the way I've heard it explained is that when you have like rain, or say you did have a really high humidity, vapory air, that gets condensed on the leaves, and then the tree structure through it kind of, if it doesn't fall in the drip line, it actually can move down the wood and down the stem into the soil, where then it would be absorbed by the roots. I think, but it's a good question that I don't know if I know for certain.
Sure. That's great. Thank you.
Mm-hmm.
And then one more related. We mentioned quite often that some trees have tap roots,
Yeah.
and we show these photos that have roots that are spread out quite wide. Can you explain the difference between I guess, the two differing but related opinions?
Well, so there will be species like that come to mind with those tap roots. I don't know. It would be a good distinction to make to say, hey, this is like what a maple looks like. And then next to it have an image of here's what a black walnut looks like, or here's what a shagbark hickory looks like. This was my first season ever planting a bare root shagbark hickory, and I don't know if I could tell you where the stem started and where the roots ended. Like it was just like a big carrot.
Okay.
But I think you potentially you could have, tap roots that can drop down 5, 6, 7 feet potentially, right? Until there's no oxygen diffusion but I think they would still have these other roots out here, right? So there would be maybe a tap root, but then still roots, lateral roots forming up in the upper part of the soil.
Awesome. Thank you.
Mhm. I'll also say it's kind of a challenge to study roots because, to actually imagine having to excavate and cross-section a column of soil to actually see the roots. So maybe that's part of a little bit of the lack of clarity on is just how challenging it is to actually observe roots in the soil. They've been trying some things like ground penetrating LiDAR and things like that to image it, but probably still some more interesting science to be done there. So I'll keep moving here. So the next couple slides, I just want to talk about trees reacting to external stimulus. So there's a bunch of different tropisms within trees. For example, gravitropism, phototropism, water tropism. So in terms of trees and gravity, if a tree is growing at a lean, it's going to have a tendency to want to grow straight. So even if you have a stem that's kind of leaning and then you see it from a distance, the top of the tree is actually straight up in the air. And this is done through the production of reaction wood, what we call reaction wood. So here you could imagine this one is, this sample is growing on a slope somewhere, or maybe it's being hit by prairie wind. And so the pith is here, but off center to the bole of the tree. The true center would be somewhere here, but it produces wood in reaction to the external stimulus. And similarly to phototropism, auxin plays a role in this. So auxin is a plant hormone, plant growth hormone, that is sort of a signaling molecule to cells to do stuff, to turn on certain genes or grow more, grow less, grow like this. So in phototropism, you can see auxin has a tendency to move towards the shade. So it's getting hit with sun, the auxin moves to the other side, but auxin then signals to the plant to elongate your cells. And then because you're elongating just on one side, it's going to push it. And so now you're having the plant lean in towards the sun. Now, once it does this for a little while and say it keeps going, eventually that auxin is going to get pushed out of those cells, both by potentially the sun or gravity. And then you could imagine it's then going to signal these cells to now you elongate. And so it's going to kind of right itself over time through these mechanisms. Okay, so I wanted to go back again to thinking about trees as energy systems. So we've got photosynthesis happening, and we need that photosynthesis. We need those sugars to do cellular respiration. We need all those sugars to build our new parts. And then we also have to keep, so you can imagine how trees need to produce more energy than they utilize, and so that they can then store it when the sun isn't shining, because they need to always be respiring. So if the energy demand of the tree as a whole is greater than what is created through active photosynthesis, and then also stored, trees are going to go into decline. And I know that using decline in this way might be a little bit controversial if you're a plant pathologist. I know there's specific decline diseases where there's like a pathogen involved, but in this context, Paul Mannion's decline spiral, thinking about decline as a successive set of stressors leading towards death. So we kind of start on the outside and we're spiraling in towards death here. So starting with predisposing factors like the site conditions, what's the soil like, what are the actual genetics of the tree. I kind of think about this as things we can't change. We can't change, if we're just a tree, we can't change the soil we're growing in, and we got the genetics that we got. So that's our current state. And then you start putting stressors into the system. Call this an inciting factor. So say I'm a red oak, I'm growing in a sandy soil, and now I'm trying to grow through 2 years of intense drought, which we've been experiencing here in Minnesota. Not this spring, it's been a great spring so far. But so now we're stressed, right? So now we're not producing as much sugar, and we've got to keep all this plant material alive, and we don't have the water to do the photosynthesis. And okay, now put something on top of that, another contributing factor, something like a 2-line chestnut borer, right? So now I've got an insect that's disrupting my vasculature and making it more difficult to get the little sugar I can make down to the roots. And so you can see it's just this little jab punch, tree death. I thought a nice illustration of this is a study that was done back in the 1980s looking at winter storage of starch in white oak roots. So you could take a root and you cut the root and then put Lugol stain. So it's a stain that stains starch. So the more stain you have, the more starch you have. So in this experiment, they found that the trees with the lowest amount of stored starch during the winter had the highest incidence of 2-line chestnut borer infestation. So these were trees that did not have 2-line chestnut borer, but then got it. And then they did this extra component of the study where they actually girdled, or they actually broke the phloem so it couldn't get down to the roots. You can see that was a very significant increase in beetle damage. So there's something about these native borers and native trees, these agrylus borers, bronze birch borer, 2-line chestnut borer, they're sensing potentially this sugar or energy or something in the tree that tells them this is a easier tree for me to infest. And then here on the right is just another example of low starch, high insect damage versus high starch and low amounts of insect. And so it's good to remember with the starch that it changes throughout the year. So looking at the red dotted line here, this is our starch reserves within the tree. And so as trees are coming out of dormancy, they're needing to use those stored starches to push that new growth. Remember, I got to push those new shoots and leaves. And then at some point, okay, now it's doing its thing and I can start saving starch again for the winter. So the decisions that we make, or in terms of taking care of trees or other tree issues, well, let me say it just like this. The tree issues which contribute to disruption or reduction in the efficiency of energy creation through photosynthesis will lead to tree decline. So here, for example, stem girdling roots, this is going to increase the pressure required to pull water from those roots up into the canopy. And so you have a situation where this tree was planted too deeply, and the roots, because they're not getting that cue from, and they can kind of just grow where the growing's good, they end up growing right up next to the stem. And the stem tissue and the root tissue can't fuse together. So you just end up with this point of compression where they're both going to want to keep getting bigger and not go anywhere. So this will lead to, because you're increasing that water pressure demand or requirement, you're going to start to see canopy decline occurring. And then this also, because of the lack of water, and then this also creates a point of weak structural weakness where you actually get trees just breaking right at these points. But that's probably another talk. Another energy requirement or demand is compartmentalizing decay. So I think we're not going to go deep on code it today, but the compartmentalization of decay in trees. Trees don't heal, they seal. And so here's just an example where there was some sort of wound out here, and then the discoloration represents decay, some sort of decay fungi probably that got into the stem. And so these decay fungi want to get at that cellulose in the plant fibers, and so they start eating and degrading the wood in that way. And the tree doesn't have any way to, sort of like our immune systems, it can't go in there and kill the fungi and then rebuild that structure. It needs to just seal it up, compartmentalize it. And so here's what it's done. It's been able to wall off the pathogen and prevent it from moving deeper into the tree. And you can see it's even trying to grow and reconnect this cambium vasculature on the outside. Another good one to keep in mind, if you're ever doing some pruning or wondering, what's the right amount, what's the right dose, right? Pruning is also wounding. So here we have an example of a Quercus bicolor swamp white oak growing on St. Paul campus, and it had to be pruned up, probably for access. You had to get above this sign. And looking at this, it's something like about 50% photosynthetic to just the total height. So we call this the live crown ratio. And for deciduous trees, it's nice to keep this at least at 60%. So more canopy means more photosynthetic capacity to support those roots, to support that stem. Another one, so we've been talking about this importance of water transport in trees and oxygen diffusion. And so when you have things like construction happening, you often will get a lot of compaction happening around the trees, just because it is what it is. You can see here we've got a dumpster parked in the root zone of a tree, and trucks being parked all day, and other building materials. On the right, even a more extreme, where this fill is just piled up. I don't know how tall that is, 15 feet or something like that, 10 feet, 12 feet. And here you're not only getting compaction, but you're also completely suffocating this root zone. So hopefully this isn't going to be here for too long, but the compaction, so in that case, the water could actually get in, or oxygen get in, but that compaction is going to be more difficult to remediate, and it just is very difficult to remediate. And so you can try, if you have compacted sites or you want to do something good, right? So if we have good aerated soil that has good organic matter, good good nutrients, then we're going to produce more good stuff for photosynthesis to happen. This is just showing using an air knife. So this is compressed air to blast out compacted soil, radial trenching, so making some trenches with the air knife, and then backfilling with a nice rich compost. You could do this whole area if you really were going for it. There's also vertical mulching, where you make a bunch of kind of holes using the air knife, and then same thing, backfill it with compost so that those fine roots can grow in there. And then also things like decay fungi. Remember, there's root decay fungi out there, and if you're degrading the roots of the tree, that's going to lead to decline of the canopy, because you're losing that ability to pull water up. Here's just an example of armillaria, which is decaying these roots, and then eventually will make its way into the trunk of the tree as well. So over time, trees will what we call retrench. So you have this kind of life stages of a tree. And so it starts small, it's growing, it's accumulating more and more resources, it's really starting to thrive. And then here, life stage 7 sort of shows it at its maximum peak, right? It's got all the energy it needs, and it's really healthy. However, trees, remember, they're putting on these annual rings, and they kind of need to keep growing. And so that there's more and more tissue to support and keep alive, which requires sugars for respiration to occur and water for the reactions to occur. And then that's without even bringing into play other stressors from the environment. Over time, they just lose the ability to make enough energy to keep up their fullness. So they'll actually start to kind of retrench back in, or you start seeing dieback from the tips, and they sort of will come back down. And and this is natural. We just do the best we can to keep our old trees alive. So I just am gonna finish on this slide, and then hopefully there's some questions, or we can just chat a little bit more about topics. But I'm hoping that I'm communicating this conceptual framework for understanding trees, that from this energy creation, storage, and utilization kind of standpoint, and then thinking about why is this tree in decline, thinking about this energy production, and what points in that chain of events are things going wrong? Is it in the shade? Was there construction damage? Is the soil, are we in a drought? And then it all kind of comes back to that photosynthesis and capacity. But Nick, maybe I'll stop there and see if anybody has any questions in the chat, or if you have any thoughts.
Sure thing. Now, because we're towards the end, I have a question for you, Ryan.
Mm-hmm.
It may prompt another webinar here, but as it relates to energy storage, starch, all that in the tree branches, how do our current best practices for pruning affect the overall health of the tree, or is the energy stored in a branch just for that branch? And so by removing it, you've taken away that branch, but you're not harming the tree as a whole. And what are your thoughts on that?
I actually was thinking about that too. I think that would have been a great slide to put in here. So for example, there's this 1-to-3 ratio comes up. So thinking about the size of the branch you're removing compared to the next branch that's going to take over, or back to the main stem. So we would say generally, if you're making a pruning cut back to the stem, you don't want that branch that you're removing to be more than 1/3 the size of the stem. So 33%, and then smaller is even better. And you could think about it as well in terms of reduction pruning. So say you're pruning back to a lateral, you would want that lateral that's going to remain there to be at least 1/3 the size of what you took off, because that branch, that cambium and that vasculature in the branch needs to be supported by the photosynthetic capacity. But even beyond that, I think the trees have this sort of, it's kind of this emergent fractal pattern to it. So yes, it needs that direct photosynthesis to support that branch, but that branch is also supporting the main stem and supporting the roots, right? Because there's vessels that come up from the roots all the way up to that stem. So if you lose those, so I think it's another interesting talk. I think just think about pruning and tree health. But yeah, thanks.
We've got a couple of questions I think we can roll into one. Part one is, when do trees become carbon neutral? And roll that into your thoughts about trees as an option for long-term carbon storage.
Okay, when they become carbon neutral, does that mean in terms of like it took a bunch of fuel to like get it in the ground?
Yeah, and that would be my understanding.
Okay, and it's a good question. I don't think I have that piece of knowledge, but we would say that it's the big trees it's once they get to maturity is when you start getting these ecosystem services returned to you. Yeah, we could do the math. Oh, sorry, and could you repeat the second part of
and just your thoughts on trees as a long-term carbon storage option.
Right. Yep. Yeah, I think that's, I mean, it makes sense, but I also just think about how decay fungi are going to be in there. As you get old trees, they start to hollow out, right? So you need to, trees are this great, you know, biomachine to get carbon out of the air and put it into a stable structure, right? Into that sugar, into that cellulose. But nature wants to break that down and release it back out. So you would have to do something with the wood. I don't know what that means. Does it mean burying it? Maybe make houses. Like, are houses actually carbon storage? But yeah, I don't know for certain. I just know that there's other organisms out there that are just ready to release that back into the atmosphere.
Sure, sure. Just as an aside, there are some really cool forest products. Mass timber is making its way into the popular world, which is very cool. There's another product out of Oregon called cross-laminated timber. It's essentially super strong plywood. You can build structures out of it like giant Lincoln logs. It's pretty cool. You kind of touched on this when you talked about stem girdling roots, but we know vines often climb up and choke out trees. So when they, certainly when they wrap around the trunk, but when they wrap around branches, does it essentially do the same, and it creates this pressure point that causes the tree to expend more energy to get everything out to that branch?
Yeah, I think there's one that comes to mind is roundleaf bittersweet. We get that up here, and it just is, to see it just strangle a stem is really something to see. But I think that's exactly how I would think of it, is that vine is trying to, either if it's actually trying to tighten down, or it's just because the branch gets larger and it's there and it's getting larger, and then you get that compression. But yeah, anytime you're going to be compressing those vessels and increasing the pressure there that's needed to get it through, it's gonna, yeah, just make it more difficult for water to transpire through there. And I should mention that most of the water that goes into trees is for transpiration. Something that I've heard, 95% of the water that ever gets taken up by the tree is just going up and out. You just have to keep those columns going. And, you know, there will be some water that gets stored as the structure gets built out, but really it's just, it needs to move that water through and keep the cycle moving.
Lost it here. Oh, here we go. Bob asked, kind of, how is reaction wood created? Great question.
So reaction wood, and it's slightly different if you're a gymnosperm or an angiosperm. So like for example, a conifer, if it's, say it's leaning, so the auxin is going to go down. It's through the auxin and through gravity. So the reaction wood in a conifer, it actually creates more lignified wood, if I'm understanding. If we're looking at this image here, so say this was leaning to the right, and so conifers are going to put more lignified wood here to try and kind of prop it up, versus angiosperms, which are going to put on more highly cellular, like more cellulose. And so there's this difference between compression wood and tension wood. And so if it's leaning this way, the tree puts on wood here, more cellulose, and then it's actually trying to kind of pull it back up. But I think it's through that auxin signaling that I don't quite have my head wrapped around yet. That just tells the cells to grow in a different kind of richness of these different molecules.
As far as young trees, sometimes we frequently see those planted with those opaque white tubes.
Oh.
What does that do as far as stimulating tree growth?
These are really good questions. It's like every time a question is like, oh yeah, I was going to put that slide in there. So there is, on young branches and stems, the bark is actually photosynthetic. So you can have photosynthesis occurring, and I have seen a photo that was sent there. The tree had been tubed with one of those opaque tubes, And there was actually like, it got taken off and it was skinnier down low, and then it opened up and got bigger. So I would recommend with young trees not using an opaque guard. You could use something like hardware cloth would be an example, or some sort of reinforced plastic mesh tube. You could go with something like a grow tube. But yeah, the young bark is photosynthetic.
Talking about EAB, do you expect the pest population to decline anytime soon? Are there advances in creating resistant ash trees? Anything of that you can speak on?
Yeah, so one route would be to go with planting non-native ash trees like Mancana ash, Manchurian ash, but it's the same system that I was talking about with two-line chestnut borer. If you have stressed Mancana ash, emerald ash borer is going to think that's a desirable tree to go after. I think we'll probably see this. It'll probably not be going away because there's lots of ash regenerating in unmanaged landscapes in forests. And so sure, it might kill off enough ash where its population will dwindle, but then the ash grow back and the remnant ash borer then grow back too. There's a group, there's a research group with the Forest Service out of Ohio that also does Dutch elm disease screening or Dutch elm disease resistance breeding, but they've also found some survivor ash trees down in Ohio forests, and they're starting to clonally propagate them and then artificially, put EAB on them, and they are finding that they do have some mechanisms. It seems like our ash trees, it's like they have the mechanisms to produce the compounds that are going to negatively affect the larva, but there's a research paper that if you put, I think it was jasmonic acid, so if you put jasmonic acid on one of our native ash trees, it actually upregulates a set of genes and compounds that then help it fight back against emerald ash borer. But for whatever reason, it's just not. So it's got the mechanism, it's just not getting the signal that there's something there. So it's going to be a long time, unless GMO stuff takes off and people are okay with GMO trees, and you just have the tree express some gene from some other plant and kills the larva.
And then I think we got to all of our questions here, and just to kind of round it out, and because it piqued my interest, can you give us a snippet or the Cliff Notes version of your work with American elm?
So we've got, if anybody's planting American elm out there, in the trade, there's a few American Ulmus americana resistant varieties available, things like Valley Forge, Princeton. Our research program here started with the identification of the St. Croix elm. So St. Croix is propagated by Bailey Nurseries, and it's a native tree growing in Afton, Minnesota. And the arborist that was taking care of the tree noticed that it would flag a little bit, but then it would keep growing, and then it would flag again, but then so it was able to kind of compartmentalize the fungus in the tree. So what we do is we go around, we find these big trees that have been here, hopefully at a fairly mature size when Dutch elm disease wave came through in the 1970s, and then propagate them clonally. So collect some dormant tissue, graft them in the greenhouse, or try and make a rooted cutting. We grow them in our nursery here for a number of years to hopefully a good size, and then we isolate the Ophiostoma, the fungus that's causing the disease, and we make a spore suspension and inject that into the stem of the tree, and then observe to see what kind of wilt reaction happens in the tree. And so we have found a few of these around Minnesota that will get injected with the fungus and then wilt a little bit, but then be able to keep growing. And so that's a real goal, and we're starting to do some reforestation plantings with these varieties that we're finding out in the landscape.
It's very cool. Very cool. One last one, because it's a hot topic. It's happening now. We have the cicada double emergence.
Oh, I've heard of this. I don't know. I don't know my cicada biology, but what would be the question about?
Any concern for established trees?
I'm sorry, I don't know the cicada life cycle, so I can't speak to it. Would you have any thoughts, Nick?
I think it's largely dependent on your location. We get cicadas every year. Their ovipositors do make small holes in small twigs. If the tree is small enough, yeah, you could see some damage, maybe some death if it's really, if there's a heavy cicada population in that space and they all choose your tree. If it's really a concern and it's possible, I would net your trees, fruit trees in particular, but it all just depends on how many come out of the ground in your space.
Okay.
I think that's all the questions that we have.
Okay.
Anything else you want to throw our way? And if not, we're at an hour. We're good.
Yeah, no, I think thanks for the idea and the opportunity to kind of think through this. I'm not really sure if that was the direction that was useful, but I thought it was interesting to think about the energy component of this and everything. So I thank you for letting me work through this, and hopefully everyone out there had a nice Wednesday afternoon. Wednesday? It's Wednesday.
Yeah. Yep. Awesome. Yeah, it was a really fascinating topic. Thanks for taking on the task. Yeah, this was great. Thank you.
Yeah. Good luck with the rest of the series and your growing season down there.
All right. Thank you very much. Thanks, folks. If you have any other questions, feel free to shoot us an email. I'm always happy to chat. We'd love to come out and visit. We'll stay in touch. Thank you, everybody.